Good morning, everyone. Hope you all are doing well. We are doing fine. I would really like to hear from all of you whenever you have a chance to let me know how is everybody doing. Okay, so moving to today's lecture. So we have been talking about DNA recombination. We talked about two different ways of DNA recombination, homology-directed repair and site-specific directed repair. Now the third way of DNA recombination is transposition, which is not in true sense a recombination, but it changes the DNA, it changes the genome of the individual because the transposable elements can move from one place to another place. So we are still working on chapter 12. The page numbers are noted here. Okay. So transposition, what is transposition? Transposition is a specific form of genetic recombination that moves or allows this movement of certain genetic elements from one DNA site to another. These genetic elements that we call transposons or jumping genes, these can change their positions within a genome, and that the change in the position then can create either it can create a mutation, or it can also reverse a mutation if there was a mutation there. So ultimately what happens if it's a replicative transposition, it will change the genome size. If it's not a replicative transposition, then the genome size will stay the same, but the genome organization will change. So here we talked a little bit about it in previous lecture. Transposition was studied by many people, of course, but the major credit goes to Dr. Barbara McClintock, because she worked on maize or corn to understand why there are so many different kernel colors happen, and this kernel color is pretty random in maize, and she realized, or she learned that this is because of the jumping genes, the mobile elements that move from one place to another place in the genome. She was awarded Nobel Prize in 1983 for this discovery. So these mobile genetic elements or transposable elements or transposons, because of their movement from one place to another place, we see these different colors. But corn is not the only organism or only plant or a plant that has transposable elements. All organisms have transposable elements, and transposable elements make a large portion of our genome. So here, let's watch this video. It's a short video. See if I can play it. All right, it doesn't look like I can, but if you are able, oh, here it is.
I'm Dave Miklas of the Dolan DNA Learning Center of Cold Spring Harbor Laboratory. Transposable elements, or transposons, are the so-called jumping genes that can move from one chromosome spot to another. First discovered here in the 1950s by Barbara McClintock, transposable elements are now known to be the most common feature of plant and animal chromosomes. Scientists have long wondered about the effect of all this jumping around. However, it has been very difficult to catch transposons in the act of jumping. Most are, in fact, molecular fossils, meaning that they jumped to their present positions millions of years ago and are no longer active. Working in rice, scientists in the United States and Japan now have had the good luck of catching a transposon in a burst of activity that produces about 40 new jumps per plant. And the surprising result of all this jumping? Very little harm to the plant, because the transposon seems to be programmed to avoid jumping into active genes. In fact, frequent landings nearby to genes often increase their activity or potentially make the genes responsive to stressful conditions such as salt and cold. I talked with Sue Wessler of the University of Georgia about what this all means.
There are some plants, like normal plants, like lilies or irises, 98% of their DNA is derived from transposable elements. So what's their function? I mean, I think now with some of our most recent results and some results from other labs, they clearly have a role in diversifying the genetic material. Whether that's sort of just a byproduct, and it probably is. I mean, it's not like their purpose. Their purpose is to survive, is to get through and get to the next generation. And the way they do that is they have to keep their host alive to live and reproduce. So a good element is one that doesn't harm its host. So that's the fun. That's the fun part about studying elements, because what we're studying are successful elements. And successful elements act in very subtle ways.
So transposable elements are almost perfect genetic parasites, silently using the chromosomes of other organisms to perpetuate their own kind. As a byproduct of the jumping, it appears that transposons may even help their hosts and themselves to survive tough times.
So that was interesting, and we will get back to this active and inactive transposable elements and how transposable elements regulate gene expression. So one of the ways these transposable elements can regulate gene expression is by inserting in the regulatory sequences of the genes. So we will talk a lot more about regulatory sequences in the later chapters, but think about it. When a DNA is replicating, or if a DNA is transcribing, that's where your DNA expression, gene expression, will occur, as the transcription of DNA into mRNA, so that can be then eventually translated into proteins. So during this transcription of mRNA, the regulatory elements on the DNA will regulate when to transcribe, how much to transcribe of that mRNA. But if transposable elements inserts itself close to that promoter region, close to those regulatory sequences, it can change the expression of the resulting mRNA. Right, so transposable elements, as we talked about these transposable elements, say for example here in this example, this is genomic DNA. This is where transposable element is. Transposable element can move within a genome in two ways. These can move without a duplication. So this will be excised from the old site and will be inserted into the new site. So now the net change here is no effect on genome size. It's affected the genome structure, but not the genome size. Another way of transposable elements movement is by duplication, where the element duplicates itself and then it will get inserted into a new site. This leads to a net gain of genome and DNA, or genome size increases. Transposable elements are the most common source of new mutation in many organisms, because how the mutations happen in the organism is by change in DNA sequences. And this DNA sequences, if this is a harmful mutation by, say, example, radiations or chemicals, the cell DNA will, or the cell machinery will try to fix that sort of mutation. But transposable elements, because they are part of the cell machinery itself, that the part of the genome, these can move from position to position and carry out mutations, will bring mutations into the DNA without because these transposable elements can insert themselves at random locations, because that's what we know right now, is that there is no specific DNA sequence required for transposable elements to insert into a new position. So since it's almost random insertion, these can be used as mutagens or DNA delivery vectors. And that's a very common way of making transgenic animals, is by using transposable elements as DNA delivery vectors. These transposable elements, as I mentioned, these are present in genomes of all organisms. And these transposable elements make a large portion of genome. More than 50% of both human and corn genomes are composed of transposable elements. And as we heard in the video, Dr. Sue. She mentioned that in some plants, up to 98% of genome is composed of transposable elements. If you think about it, in human genome, it's about only 2% of the genome codes for proteins. The rest of the genome is either transposable elements or repetitive sequences. So, transposable elements, we can divide these into three major families: DNA transposons, virus-like retrotransposons, which are also known as long terminal repeats or LTRs, and poly-A retrotransposons or non-viral transposons, non-viral retrotransposons. So out of you'll see here there's one DNA transposon, and then two of the family members are both retrotransposon. That means these are RNA sequences that need to get converted into DNA by complementary DNA synthesis by reverse transcriptase. These virus-like transposons are what the video in the video Dr. Sue was talking about, that these elements have successfully integrated themselves, or these could be the molecular fossils, as they said in the video, because these virus-like retrotransposons were believed to be viral DNA that have been integrated in the eukaryotic chromosomes over the hundreds of thousands of years, and have eventually lost their potential to be viral genomic DNA because they don't anymore code for the proteins that are required for capsid. So they stay, so they cannot cut themselves out, so they're not go into lytic phase, so they always stay with the genome. So they became eventually the part of the genome. All their characteristic resembles to viruses, RNA viruses. So that's why these are called virus-like retrotransposons. Poly-A retrotransposons are a very different class, and we will talk more about those later. But these are called poly-A retrotransposons because, like mRNA, these retrotransposons also have a poly-A tail. So they are structurally very similar to coding genes or mRNA sequences. These are the examples, the schema of all these different transposable elements. So here the first is DNA transposon. These DNA transposons have this is the element. It starts from this green to green. This is the complete element, complete transposable element. Now these transposable elements contain these terminal inverted repeats. These terminal inverted repeats are very important, as you will see later. And this sequence here that will code for a protein for an enzyme, transposase enzyme, that's an endonuclease. Okay, so the DNA transposon, this complete element consists of terminal inverted repeats on both ends, and the sequence that will code for transposase. These in blue, this target site duplication, these are part of the host, part of the DNA that they are inserted into. So wherever these transposable elements insert themselves, because they have to break the DNA there, it usually adds a couple of nucleotides when the repair mechanism is fixing those gaps, and these target site duplication could be as short as 2 base pair. It could be as long as 20 base pair. These target site duplications are usually an indicator that a transposable activity has happened in this portion of the genome. This is the flanking host DNA. That's where this is present. This virus-like retrotransposon or retroviruses, which is now we are dealing with one of the retroviruses, COVID-19. It's an RNA virus that's structurally similar to these virus-like retrotransposons. The difference you can see between these two transposable elements is, again, the blue is the target site duplication. These are not part of the element, but these are the portions where the transposable elements insert itself in the genome, and these small portions get duplicated. The transposable element itself starts from here, from this green to and ends at this green. These retrotransposons have this long terminal repeat. So here you can see these terminal repeats are long here. That's why they're also called LTRs, or long terminal repeat transposons. And then they also have the sequence that will code for enzymes, code for these proteins, integrase, which is a transposase, and a reverse transcriptase, because these are retroviruses. Their RNA sequences, these need to be converted into DNA in order for these to get into the genome. So the DNA sequence get converted into RNA, back to DNA, and then it inserts itself into the new position in the genome. Poly-A retrotransposons, as I mentioned, these are very similar in structure to mRNA sequence. They have poly-A tails. They do not have these terminal repeat ends. Instead of these terminal inverted repeats, they have UTRs, or untranslated region. 5' UTR and 3' UTR. So 5' untranslated region and 3' untranslated region. In addition to these UTRs, they have two ORFs, or open reading frames, ORF1 and ORF2. These ORFs code for integrases and reverse transcriptases and RNA binding proteins. Okay, DNA transposons, let's talk more about these. They have a DNA sequence that function as recombination sites and gene encoding proteins that participate in recombination. Let's just look at this here. These are those recombination sites. These green are recombination sites, and these are the genes that encode for protein that participate in recombination or that make a transposase. Recombination sites are on both ends. These are inverted repeats. These are organized as inverted repeats. These can be less than 25 base pair or up to a few hundred base pair depending on the transposable element. This recombinase is responsible for transposition. It can be known as integrase or transposase. This is an enzyme that will bring these molecules together, the inverted repeat sequences together, and make a cut. Target site duplication, these target site duplication over here, these are 2 to 20 base pair direct repeats. These generated during recombination, but these are not part of the mobile element. So here again, this shows the structure. You probably now know it pretty well. We don't need to go into too much detail. Again, the target site duplication, the element starts from here and here, inverted repeats, transposable element, transposase coding sequence. Okay. Most DNA transposons are autonomous transposons, which means that they carry a pair of terminal inverted repeats and a transposase. So therefore they have everything they need to promote their own transposition. So here, if the element has both inverted repeats as well as transposase, it's an autonomous transposon. But some transposons lack the sequence that codes for transposase. They will have inverted repeats, but no sequence that codes for transposase. In that case, it's called non-autonomous transposon. These non-autonomous transposons are those molecular fossils that the video was talking about. So these cannot move from one place to another place because these cannot, they don't have all the elements that are needed for their movement, for their transposition. However, these non-autonomous transposons can move if there is another transposable element, a helper transposable element that has a sequence very similar to these transposons. So unless there is an autonomous transposons that's very similar in sequence, these non-autonomous transposons, they remain frozen, they're unable to move. One of the good examples of non-autonomous transposons is Sleeping Beauty transposon. The Sleeping Beauty transposon in many insects is called Sleeping Beauty because it has been there for a long time. It's a non-autonomous transposon. It doesn't move. But if you use it, this can be used as a molecular tool where if you inject a very similar sequence with a transposable activity, you can move the Sleeping Beauty transposable element from one site to another site. And so you can attach your own sequence with this transposable element, so wherever the transposable elements land, it will also bring that sequence with it. Okay, so again, this is an example of an autonomous transposon. If it doesn't have this portion, then it's a non-autonomous transposon. And many transposable elements in our genomes are non-autonomous, because if there are too many autonomous transposons, then there will be a lot of genetic instability happen because of activity of transposable element. But since these transposable elements, many of these transposable elements lack their ability to transpose, then they just stay in the genome wherever they are without almost always moving around. Okay, so here is an example of how transposable elements are used in the research labs as tools for transgenic development. In this example here, this is an example of Drosophila melanogaster, the fruit fly. So here in this example, we have these females with white eye. So this is the genetic background. Does the fly have a white eye? This white eye gene, this is the white eye gene. So if you insert your gene of interest in this white eye gene, then what will happen is, if this gene is inserted there, that will disrupt white eye gene. What happens is, when white eye gene is disrupted, it gives the red eye color to flies. So in this example, you made this promoter sequence. You made this plasmid with white eye gene with your gene of interest inserted in it, and then you have this transposase, a helper plasmid that contains a transposable element. You inject both these plasmids into the embryos of this G0, this fly. This is a white eye fly. She laid eggs. You took those embryos, you injected both plasmids together, the helper plasmid as well as your transgenic that you want to make, that the gene of interest you want to insert. Once you injected this, you will look for the individuals that have mosaicism in their eyes. For example, this individual here has mosaic pattern. Well, this mosaic pattern means this is the expression is heterozygous. It's not a homozygous expression. That means not both alleles have been changed, only one has been changed. And you take these mosaic individuals and backrow those with the wild type, your white eye individuals. Now, according to Mendelian inheritance, some of these will be white, some of these will be mosaic, and some will be homozygous. Then those homozygotes will have all red eye because they didn't get any white eye. And here you will select for those G1 red eye color transformants, and to achieve this transformation with transposable elements, we have to inject a tons of embryos though, because the transposable elements are random. So not all of your transposable elements will land where you want it to land, close to your white eye gene. So they might land somewhere else, and it does nothing. So the efficiency is about 1 to 2% usually with transposable elements. But once you get the homozygous line, then you can rear this homozygous line, and you will have this line, so that you will have a stable germline transformation. Okay, so now let's move on to the next class of transposable element, virus-like retrotransposons, or long terminal repeats, or LTRs. These virus-like retrotransposons, they convert RNA into DNA, and that's the reverse of the normal central dogma of biology, normal pathway of biological information flow. That's why this is called retro. These carry, similar to DNA transposable elements, they also carry inverted terminal repeats, but these inverted terminal repeats are a part of the larger long terminal repeats, so they are within this long terminal repeats. These are the inverted repeats, these are the long terminal repeats. These are embedded within these long terminal repeats. Okay. These virus-like retrotransposon, these encode two proteins that are needed for their mobility, integrase and reverse transcriptase. Integrase is required for the transposable activity, and reverse transcriptase is required from RNA to DNA synthesis. These retrotransposons, they act like retroviruses. They are very similar, and they potentially those were retroviruses a long time ago, before they got inserted and just stayed there in the genome over millions of years. They just stayed there, became the part of the genome. They act like retrovirus, but there are differences between retroviruses and retrotransposons, and the major difference, of course, is that retroviruses are packaged into viral particle. They escape the host and they can infect the new cells, and that's what viruses do. But retrotransposons, they cannot package themselves into any sort of particle. They do not escape the host, and they cannot infect a new cell. They just always stay within the same DNA. They can move from one site to another site in the same DNA, but they will never leave the cell because they're not viruses anymore. Okay. Poly-A retrotransposons, they look like coding genes because they have the signature of what the coding genes look like. They make mRNA. They do not have these long terminal inverted repeats like retrotransposons, the other virus-like retrotransposons. Instead, they have these two ends of the elements. They have distinct sequences called 5' untranslated region and 3' untranslated region. These are followed by a poly-A sequence. Both of these UTRs are flanked by short target site duplication, but that's common in all three classes of transposable elements because this is part of the host DNA, so that's where it's going to insert themselves. They usually add, it leads to addition of a few base pair, and that's what this duplication is. These poly-A retrotransposons, they carry two genes, open reading frame 1 and open reading frame 2. ORF1 encodes an RNA binding protein, so this ORF1 here encodes a RNA binding protein, and ORF2 will encode the protein that encode for both protein that is required for reverse transcriptase and endonuclease activity. Again, these can also be autonomous or non-autonomous. That means that if they have all these elements, they have ORF1, ORF2, and their UTRs, and their poly-A tail, then these will be autonomous. But if they lack any of these, this portion of their element, then these will be non-autonomous. So that means these can be activated, they can move from one place to another place in the genome, or they can be frozen, stay in the same place in the genome. Okay. So let's talk about transposase mechanism, how the transposable elements move themselves from one place to another place. So one of the common mechanism is cut and paste. All three classes of transposases use this mechanism, this cut and paste mechanism. So here, see, this is an element in old DNA location. This is an example of DNA transposable element. This is the flanking host DNA. These are the inverted repeats, and this is the central portion that will code for transposase or integrase. Right? So this coded for these transposases or these integrase bind to these inverted repeats. They make this synaptic complex. Okay, this is the simplest transposition reaction is the movement of DNA from one site to another site without replicating the transposon, and that's what cut and paste is. So it's cut from one site and get pasted into a different site instead of copy and paste. So what it does is it's the excision of transposon from its initial site to the host DNA in the host DNA and integrate into new site. So cut and paste transposition. So this makes these integrases now bind to inverted repeats, and this will then lead to the excision of this transposable elements, and let's talk about that in more detail. To initiate the recombination, these transposes they bind to the terminal inverted repeats. It brings these two ends of the transposable element together, makes this DNA protein complex called synaptic complex or transpososome. This transpososome is very important because that's the first step in cutting it from the existing DNA sequence. To excise DNA, these transposase subunits within the transpososome, they will first cleave one strand on each end of on the transposome. So here it shows the cut on both ends, but that's not what usually happens, and we will see that later. So here, this is the new DNA molecule. This is the transposable element. This transposable element cut itself in a way that both 3' ends have hydroxyl groups. These hydroxyl groups can make a break in DNA, so they made this break in the DNA. Once the break is there, the transposable elements will come and bind, and here then this break leads to this repeat element. So this sequence will be repeated, and that's what those direct repeats are. DNA repair mechanism now will fill this gap, and now this is ligated. This is the new DNA sequence that has been inserted here. This is target site duplication. So this is, we will be talking in a little more detail. So this transposase cleaves the DNA in a way that, as I said, that the free element has a 3' hydroxyl group. These 3' ends of the transposon liberate first, or here, this is a better example of what happens. The transposable element makes this cut at 3' ends. So these 3' ends liberate first. The 5' end is still joined with the host DNA. Now, different transposable elements have different ways to cut the second end. In some transposable elements, this hydroxyl group here will make the cut on this end as well, and this we will talk about that later. In some transposable elements, it's a different set of enzyme, a different enzyme that comes and makes that break. For example, in TN7, so here, so this TN7 uses this protein called TNSA. This is an enzyme that will make this break at the 5' end of this molecule. So 3' end is cleaved by the transposase activity, and a different enzyme makes this break at the 5' end. Now this molecule is out. It's both end has a hydroxyl group. This hydroxyl group will then make the break in wherever it's going to insert itself, and then it will transfer there. So this 3' hydroxyl end that attacks the DNA, and DNA phosphodiester bond at the site of insertion. It's called the target DNA, and it inserts itself into this target. Once it's insert itself into the target DNA, now the transposon is covalently joined to the target DNA, and this DNA joining reaction results in a nick. This nick will then be filled by the DNA repair mechanism by a polymerase. This DNA joining reaction is called DNA strand transfer. These two ends of transposons, the two ends of transposon, because they both have this hydroxyl group on each 3' end, these will attack the two DNA strands of the same target strand together, of the same target DNA together, and then because this is now separated by a few nucleotides, this leads to these short target site duplication. These flank the transposed copy of the element, and as I mentioned earlier, these sequences are usually a telltale sign that transposition has happened in this region. Once this happens, then the remaining recombination steps are performed by cellular DNA repair proteins. That means they will fill this gap. The DNA polymerase will fill this gap, and ligase will ligate the sugar phosphate bond. Okay, so another way, this is a way we talked about this how in TN5 the transposable elements, the TNSA protein is the one that makes the 5' end cut. There's another mechanism where it doesn't require any separate enzyme to make the 5' end cut. This hydroxyl group itself make the cut. Is called DNA hairpin. This cleaving is another mechanism, another way of cleaving the non-transferred DNA strand. It's called non-transferred because the 3' ends are the one that's usually they're the first to attack the new DNA strand. So what happened in this is the transposable in this transposable activity, this hydroxyl group will break this end here, this break here, this here, and this will then these pins, once these are both ends are cut, then the esterification reaction will close these ends. That's why it's called a hairpin, because it looks like a hairpin. This hairpin, however, needs to be opened when it finds the DNA where it wants to transfer the strand, or where it's going to. This is the host DNA where it's going to attack. Again, these are just a term, the host DNA or donor DNA. The genome is still happening in the same cell, in the same genome, within the same nucleus. Okay, so the example of this hairpin, DNA hairpin, is TN10 or TN5 transposable elements. This hairpin is then cleaved by the transposase, because the element has its own transposase sequence that will make these cuts whenever it's ready to integrate itself. And these 3' hydroxyl ends, again, these are ready to be joined to new target DNA, and these will make these breaks in the target DNA. So only difference here is that in some transposable elements, there's a different enzyme that makes these 5' end cuts. In some transposable elements, they do not require a different set of enzymes, but these hydroxyl group can make these cut, and the esterification joins these cuts until it's ready to integrate. It's called hairpin. One of the very good example of these DNA hairpins is the Hermes transposable element, or Hermes transposon. These Hermes transposon elements, have been used extensively in a number of different organisms as a tool, as a transposable element for transgenics synthesis. These are a member of HAT or HAT family. These use this hairpin to excise the transposon. This Hermes transposition, as I said, these are active elements. This is active family, head family elements. These are present in wide variety of eukaryotic organisms. So they are present in invertebrates, in plants, and invertebrates. So they all different orders of animals as well as plants have these HAT elements. There are a number of different HAT elements, though. These HAT elements are, the name HAT comes from three members or three different organisms from their transposable elements. So here, for example, the HAT, the H comes from the HOBO element from Drosophila melanogaster. The A comes from activator or AC element from maize, and the T comes from TAME3 element from Anthracium majus. So these are this is a large family. These HAT elements are active in a number of different organisms, including the vertebrates. When it's vertebrate cells, that means vertebrates. The Hermes transposon is very active in flies, and it has been used as a major mechanism other than piggyback to insert new genes. This paper is a really good paper to not only understand hairpin formation in TN5 transposition, but also the difference between hairpin formation and the other transposable elements that do not use hairpin formation. This also talks about precise and imprecise hairpins. So if you want to know more about transposable hairpin formation and transposable elements, you might want to read this paper. So it's really, again, it's a really nice paper. It also talks about this particular hairpin in TN5 that they've been studying. It leads to a 9 base pair gap flanking the integrated transposon. That means this will provide a 9 base pair of duplicated sequence at the site of transposition. So that's all for today, and then we will talk about more about different mechanisms of transposable elements and some more examples of transposase activity and their use in the research lab. So that will be the next lecture. Okay, and stay safe. Bye.